Okay, cool.
Okay, we are live, and I'm gonna wait a few seconds, and I'm gonna get walked on by YouTube's asinine goddamn user interface. There we go. Got it. Stop them. So, with Meny Karen, and we are gonna take a look at
Hey everyone, hi.
Otangelo conversation. Do we have a
Yeah.
delay? Have I been walking on you?
No, I don't think so. I think we're cool.
Okay, good. Let's see. Otangelo logical leaps, and I'm
So
sharing the screen.
even before we go into even before we go into anything,
Yep.
there's something I wanna put out there. I had like the fortune of studying BA in psychology in a really good university. And one of our courses was history of psychology. And when you study history of psychology, they gave a lot to metaphors in science. And you learned that somewhere there was this guy called, you might have heard of him, Descartes.
Yep.
And this Descartes guy, he wanted to figure out what made humans tick. And he was walking in the gardens in the I think it's the Queen's Gardens in Paris. And they had these mechanical statues that work on water, on hydroplates. So you basically step on a flagstone, and it moves fluid into the statue, and the statue moves.
Oh wow.
And Descartes looked at it and he said, "Oh my God, humans must have a fluid inside us, and something pushes the fluid,
Yep.
and that's how we behave."
Yeah.
And he came up with an entire metaphor based on this water system for humans. Now, if you move a couple hundred years forward in time, you have the steam engine, which is like the newest thing. Okay? And there was this guy who looked at a steam engine. And if you think about it, a steam engine has the fire, right? Which you have to constantly feed. And that heats everything up and creates the gases that allow you to do the work.
Yeah.
But you also need you have the chimney to let out excess gas. And that person came up with a steam engine based model for the human psyche. His name was Sigmund Freud. He called the fire the id, and the actual steam, what do you call it? It's not a container, a boiler, basically. The boiler is like the ego, and the chimney that lets out the excess is the super ego that regulates the pressure. And he just built his entire the basis for his psychoanalytical theory is the steam engine. Today, we build our scientific metaphors on computers.
Yeah.
The human brain isn't a computer. It's a sack of meat
Yep.
and fat, and it's like and biochemistry. It's not a computer,
Yeah.
but because we think in computerese, it helps us to use this metaphor to address the brain, and this metaphor works great.
Yeah.
Previous generations had their metaphors, and maybe one day somebody will come up with new metaphors for if you start confusing the map for the territory, and you think that the metaphor is the actual reality, you end up with a mess.
Yep. Yeah,
So
it's like this idea that the brain is like an electric circuit. That neurons are like a electric wire. They're not. They're more like a sluice way with a bunch of like gates that you pull up, and so the current goes a very short distance, and then another big rush of sodium ions comes in, and then
mm-hmm,
goes another
mm-hmm.
short distance, and it's nothing like an electric circuit at all. It's a
No, it's
biological
not.
entity. So,
What is a bit like electrical circuitry is like the axons are somewhat reminiscent of the idea of a cable, but it's like you said, the potential moves a short distance, and then it has to restart,
yeah, it's more like a
and then it
pipe,
moves
like a
another short distance.
yeah, it's more like a
And
sluice way or something.
yeah, until
Like
it finally gets to the end, to the synapse, and then it causes the release of more chemicals.
yeah, bubbles of
And there's
chemicals.
no central processor, there's no GPU, there's no motherboard,
Yeah,
there's no there's no RAM, there's no bias. There's not a basic input output system in there that starts everything up. That's not the brain. That's just a metaphor that helps us deal with it. Because otherwise, all we do is write equations in math or biochemistry, and we can't like utilize it. We can't uh, uh, gain anything out of it.
yep,
So that's one thing. The confusion of metaphors for reality. The second thing that's killing me is, help me out here. These people keep complaining that information has to come from an intelligence, right?
right.
So I'm asking myself, I look up at the sky, I see dark clouds, and I understand that it might rain. I have information about the weather, and I use this information to make decisions. Where did it come from?
Yeah,
What gave me the information? Well, I got it from a cloud.
yep.
Is the cloud intelligent? Is it sending a message?
Intelligently designed. Yeah,
Exactly.
yeah.
Are clouds now talk?
They have it
Yeah,
ass
sorry.
backwards.
Exactly. They see everything like you read in the Bible where uh you know how to work iron because God gave you iron working,
Yeah,
right?
yep,
And we all know that's not how it actually happened. It happened the exact other way around.
yeah,
We just started working with things and we got better at it step by step until we turned it into an art which we could pass along from one generation to the next. But um, it's the same go along.
yep.
Are
You, you bought it a little bit. Are you walking?
yeah, I'm walking around. Yeah.
Okay. Uh, yeah. You bought it a little bit wherever that was as a weak spot. Anyway, I wanted to fight with you about Michael being like meaning like God, who
Oh,
means
Mikael.
yeah, who is as God. Yeah. Well, I
Yeah,
don't know.
something like that.
I don't know if I told you who I am, but I am God. Okay, I'm sorry.
Well, if you read all of my comments to Antangelo, I try telling him that if God is everywhere, then God is in randomness as well,
Right.
and God is in the chemicals that made abiogenesis, and I don't see the problem. I mean, it, it's like I don't see the problem he sees.
Yeah.
Even if there is a God, so what? Randomness is beyond him. Oh, God can only make plans. God cannot let anything. God is like this control freak. And I'm thinking, well, maybe there's a grand plan, and then some minor details can be left to chance because it'll work out in the end, right?
Yeah, and they were saying the other day that God doesn't micromanage. That's why he allows rape.
Well, um, that's just it. I mean, if there is such a thing, it's in a universe,
Yeah.
but whatever. So,
Okay, so
I
your
think,
first comment,
yeah.
go ahead.
Hmm.
Were you done? Okay. Okay. Cells use Boolean logic. Of course they don't. That's silly. And I showed him that yesterday. Demeter emphasized that it's analog signal. It is not Boolean. It's not
Yeah,
on
but
or off. But
read what.
but we can,
Okay, read my comment. My comment isn't based on scientific knowledge.
okay.
Demeter knows the actual science. I don't.
Yeah, the problem is you're looking at it upside down. I call it ass backwards. My comment is in red there. That's not yours. That's mine. Uh,
Uh-huh.
where do we come up with logic? By observing nature. So we look at that mTOR pathway and we say, oh my God, it's turning things on and off. So we could use our idea of Boolean logic, which we invented somewhere in the 20th century, to come up with
Yeah,
an analog, And then
we
they say,
needed to
oh,
describe on and off.
right. So, so your point is that we observe nature, and that's how we come up with these ideas about Boolean logic and computers and how to build a truck and stuff like that. And they've got it ass
Exactly.
backwards or upside down. They
Exactly,
think,
because nature just does what it does, and it has a certain property,
yep,
and from that property, we derived something called logic, and then we reapply it
yeah,
to nature. But nature doesn't. Nature just is logic. If or whatever, it has its own there, and we are forcing certain interpretations on it,
yeah. And the
right?
important
Because we
thing
have to.
they want to say that intelligence like ours created life. Well, it's just the opposite. Life created an intelligence like ours.
Yeah,
So,
and I'm trying to tell Otangelo that if like what is cooler, what is more amazing, that some grand engineer designed something and it works? Okay, yeah, that's what engineers do. There's nothing interesting about that. But if there isn't an engineer and this all somehow, well, made itself is wrong, but sort of came forth, okay,
yeah.
or emerged, that is way cooler.
Yeah.
It's like we have something that is completely undesigned and yet is so spectacularly amazing, and it friggin' works. You
There's
know?
this what would you call him, rationally impaired person named John Maddox,
Oh God. Oh
who
God.
talks about biosemiotics, or
Yeah.
the application of information theory and semiotics to biology. And he thinks he understands something about information theory. I think he said something about those of us who work in information theory laugh at these atheists. Actually, I've got many books on many. information theory and biosemiotics, and not one of them talk about intelligent design. Not one of
Of
them.
course not.
Not one of them see what the rationally impaired. Well, okay, let's be fair. John Maddox was a fucking idiot. He's 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 a blathering idiot who is so full of himself. Now he may have an average intelligence, or maybe even 5 points above average, but he doesn't know how to use it. And he's
exactly.
Huh?
You know what I will say for Otangelo?
Yeah.
Otangelo, I think, has a serious issue, but at least he's not trying to pretend that he's like something he's not. And if John, if he would only let go
I know
of his insistence of using the most complicated language you can find for any simple thing, because he thinks if he says it with really, really big words.
Otangelo, last night when I showed him the mTOR pathway, he admitted easily that he had not looked at it in detail, and so he's willing to admit what he doesn't know. But
well, that's just it. When you talked about code, code is elegant.
Yeah,
code is simple. And the biological is not elegant.
Yeah, code is to the point.
Not simple. Exactly.
Yep.
If you gave instructions, and you don't need to be a coder for this, think if you're trying to give instructions to a bunch of people in order to accomplish a mission, okay, you would never do it in the way a cell does it.
Right.
If you want to say something to Michael, you just go up to Michael and tell him what you want to tell him. You don't choose this sort of random half-assed process through 100 other people who don't even need the information when Michael is standing 2 feet from you.
Yeah, there are cases when...
I tried to...
Go ahead.
Sorry, go ahead, yeah.
Oh, there are cases in code when you're looking at somebody else's code, and like you're looking at programmer B who came along and modified programmer A's code, and you're programmer C, and you're looking at programmer's B's code, and you'll see some cases where he did the most ridiculous fucking thing, where he added another if statement when he already had one, or he made an if then else, or an if then else statement way too complicated. And you can see that what he did is he just went in, he didn't really understand the code. He just went in and fixed the problem by throwing a piece of mud. And we call that a mud ball in code. You know, when people keep throwing mud at the code. And that's exactly what cells look like, is like you have too many programmers throwing mud at a problem. And then what you do is you look with intelligence, like real intelligence looks over the whole thing, and you simplify it and you find out I only need an if then else, if then and an else clause once. I don't need to nest them and, you know, bury the logic.
Exactly.
So, I mean, that's a real common thing. And that's what the mTOR looks like.
If I want to achieve something that can be achieved with an if then like with just a simple switch between two options.
Right.
I don't put a million useless, silly, redundant conditions in the way just to get to that last one. I just do the one I need. Right? I don't start 100 ifs that have no meaning. There's no point. And that's how biology works, as opposed to coding, as opposed to what we design. That's why it's sometimes so difficult to understand. It's not difficult to understand because it's so brilliant. It's difficult to understand because it's a huge mess.
Yeah.
And
the more we discover about biochemistry, the more complicated it's becoming, and the more we realize what a mess it is. Like,
Yes, and I
like
have.
we used to think they were just, genes and they produce proteins. Well, then we found introns and we said, oh my God, so there are huge spaces in between the exons that don't do anything. And we realized, well, there has to be a way to splice them. So inside of that intron, there's information to splice a protein. And then we realized, well, it doesn't always splice because it's not a perfect fit. It's like if you try to take an 11 millimeter socket and on a 10 millimeter bolt, right? Or a
Yeah,
3/8 bolt. It doesn't quite fit, but it sort of fits. It fits enough to turn the bolt all the time. But you're going to strip
yeah,
the goddamn bolt if you keep doing it. So then we realized that, okay, introns aren't perfect, and sometimes there are alternative splicing paths. So now we have a gene that produces multiple proteins. Well, then we look at the introns
yeah.
and we find out if you read them backwards, every now and then they create something called a microRNA. And that little microRNA goes out and it can attach onto another part of another gene and another chromosome and do something else. And sometimes it gets out of the nucleus and it affects transcription. It's like,
Yeah.
fuck. And then we find out that microRNAs not only do that, but they get out of the cell and they go and fucking attack other cells and tell them what to do. So, I mean, it's a bigger ball of mud.
I work at a factory, okay? My job is I operate this really, really huge machine that takes basically, let's say, nylon films and coats them with adhesive. And basically, we get self-adhesive tape at the end, okay? As in masking tape and such, right? Cool. The machine is more than 30 years old, almost 40 years old, and every which while they add another piece to it. Now, some of the parts have been added last year and are state of the art, and each of them has its own controller, and there's no main control board for the machine, because it's like everything is a different piece that's been stacked onto the basic skeleton of the machine. That's evolution.
Yeah.
And when I want to do things, I like reset something, I have to go and punch in on 5 different monitors because each station has its own monitor and there's no one control panel for the whole big mess. And that is how a machine evolves.
Well, if your factory
That's not the original design.
If your factory was like a cell, it would use that tape to feed itself, and then it would basically get really big, and then it would divide, and then there would be 2 factories. And
yeah, I know.
And
I'm saying that if you look at a machine that has been designed as it is, then of course you will have one control panel for the entire thing. Whereas this machine was added onto, and each piece is not part of the original design and is designed by someone else than the company that's going to design the other piece.
Yep.
They're all different companies, all different so it's insane. And that is what evolution looks like. It does not look like a grand design. It looks like a mess. And he tells me, Otangelo tells me that cells use quantum mechanics to communicate. And all I can think of is, well, quantum mechanics is reality. What else would they use? Magic?
Right. All chemistry uses quantum mechanics. I mean,
exactly.
They think
that's
that because,
like saying,
because they discovered a quantum effect in a cell, because they they figured out some quantum tunneling effect of the structure of some molecule. They think that an even greater intelligence who knew quantum mechanics did that.
Yes.
So we needed Leo
And
Filius.
did you know that clouds, they all have PhDs in electromagnetics,
Oh yeah, yeah. That's how
right?
they make lightning.
That Yeah, of course, because they have to use the electromagnetic force and static electricity, and
Yeah.
hey, they need to know how to. Oh wait, does that stop making sense now? Does a crystal know how to build itself? It's preposterous.
Yeah.
It's
So
like,
I have your first comment up here.
okay, yeah,
Otangelo
go
claims to
ahead.
that having seen intelligence design things, it is logical to infer an intelligent designer. And then, well, by the same standard, what does Otangelo do with the following claim? Having only ever seen natural unguided processes, it is logical to infer that natural unguided processes brought about life. And I was trying to I was trying to make that point to him that he has never. He says man has tried to develop life, in the laboratory for 70 years and hasn't been able to. And there you go. That
Yeah,
means that intelligence can't design life, right? I mean, if you use his logic, then he has seen that
yeah,
intelligence cannot design life. So you
yeah.
assume that there cannot be an intelligent designer, but he
It's
doesn't get that.
Yeah, that's making a reductio ad absurdum. That's like
Yep.
showing that if you take his logic all the way, he wrote to me that I use the same trick, that cells literally have trucks and roads
Yeah.
and GPS.
All right.
So, and then I stopped and I replied, they literally have a GPS. Cells have sent satellites into space and have built a global positioning system,
Right.
because that's what it means
To
to
guide the truck.
literally. Yeah, exactly. Because
Yeah.
if you're saying they literally have GPS, then you are saying that they have satellites in space. You are deranged. So
Yeah.
it's not literal. It is a metaphor for whatever. But of course, cells do not have GPS. Uh, uh, that can't even be responded to. Okay, they don't have roads. They have, I don't know, filaments or something?
For one
That's
thing,
saying a cell. Okay, saying a cell has a road is like saying jungles have roads, because
those
the apes use the vines. Those are roads.
those microfilaments that they're talking about, or those microtubules that transport things.
Mm-hmm.
The thing they don't
Yeah,
tell you is that those roads are being deconstructed on one end and constantly rebuilt on another, and
yep,
so they're
I know.
constantly in flux. They're constantly changing, especially in the neuron.
I, I told Otangelo, a cell, if compare a cell to a factory, then imagine a factory where they try to package their products by having 1,000 workers on one side throwing the product in the air, 1,000 workers on the other side throwing packages,
Yes.
and since they only need to sell 100 to make money, well, 100 is likely. Maybe they'll get 101. They've made their money. And hey, it works.
Yeah. So I imagine that's how an Amazon shipping center works. They have all 100,000 workers throwing 1 million packages around, and eventually they manage to ship 10,000 a day. So
Yeah,
they throw them
but
randomly around. Do you want to go any more into your comment? I was going to open this up to the
yeah,
guy.
go ahead. I think I wrote some nice stuff there. Go ahead.
Yeah. Let's see. Having only ever seen unguided processes, it is logical. Okay, we read that. I would further claim that even something as complex as a human is an unguided natural process, or maybe a self-guiding natural process, but that's a side issue. That's where biosemiotics will come into play. Importantly,
Exactly.
I would go on to claim that Otangelo too has observed unguided processes create, bring forth, give rise to, and he has never seen a new supernatural thing do jack all. So he has never seen his God do anything, nothing. He cannot even describe
Exactly.
a supernatural
And he
process.
has, just like us, Otangelo has seen natural processes make something.
Avalanches
He's
and
seen that.
riverbeds. Yeah.
Yes, or snowflakes or whatever.
Yeah.
But he has never seen, right? Go on.
He therefore cannot help but admit there's a logical basis for abiogenesis. He can still think it's wrong. We all know that logic alone doesn't always get us closer to reality, but he cannot help but understand why we find this worthwhile. But yet he does not see it. When I showed him that mTOR pathway, I could show him every single individual mechanism that would allow you to visualize what evolution on a whole does. Because we have that complicated pathway. And if we have a duplication and a mutation, or just a bunch of mutations or a brand new gene, and if it fits somewhere in that pathway, it's going to create one of those roads that go nowhere. But eventually, with enough mutations, that road, and it doesn't take that many mutations to connect those roads up. So that road that goes nowhere may all of a sudden go somewhere and actually do something. And then the cell is either going to live or it's going to die. And if it lives, it may live at basically with the same efficiency, or it may live at a higher efficiency, in which case that whole organism is going to benefit from that change. And that whole organism is going to produce 1.01 times as many offspring as the other. And over a period of 1,000 generations, that's going to take over the population depending on the size. So anyway, I showed him every single step of evolution, and that's why I believe evolution, because I know all those steps, right? And he
remind me, Mike. I'm
understands.
going to stop you for 1 second.
Okay.
I'm going to need you to remind me. I want to run by, because I know squat about biochemistry.
Okay.
And I can barely make sense of what Demeter says, which is me, not him. That's me. I have something I want to ask you regarding Demeter and his ideas, okay? Right? So let's not forget to do that. Mike, you there?
Yeah, I am, but could you repeat that? I was trying to type. I'm sorry.
No, so I was saying I want to run by some. I have this helpful metaphor I've built for myself, an analog for what you and Demeter are talking. His ideas about abio,
and you want to,
and I want to run it by you. Yeah, let's see if it.
Demeter is here.
Oh,
okay.
Yeah, if you say Demeter's name, he just appears.
Exactly.
He's like a fucking genie, you know.
He's like abiogenesis.
I'm everywhere. Just think for me.
Abio Demetrius or something. Yeah.
Think for me, and I'll be there.
Demeter, where are you from, if I may ask?
Yeah, I'm from Bulgaria.
Oh, okay, cool.
This is Balkans in Europe.
Yeah, I'm Israeli, so yeah.
Oh, you?
I know where.
Okay. And I remember, so Israeli people are very thankful to Bulgarians, because during the Second World War,
yes,
we are the only nation that actually saved our Jewish people.
Yeah, you are one of the only ones. Yeah.
So anyway, so
I'm familiar
nice
with that.
to see you. And I was listening what you said. I mean, actually, it's a wonderful. I mean, that's exactly what I'm thinking as well.
Oh, okay. So let's drop Otangelo for a second, and like, I studied psychology and a bit of neuroscience, a bit. Started working on the masters and had to stop. um, and I usually have a pretty good mind, but I don't know like chemistry, biochem, like just the real basics. Okay,
Yeah.
and I try to get more at the like the concepts and not the nitty gritty, because all the details and all that mess, that's your job. That's the boring shit. That's what the scientists do, and they save it from that job from people like me who are only interested in the principles. Um, so I was trying to come up with something that maybe I can understand. And I remembered there was a psychologist, his name was Thorndike. And Thorndike did this study with cats where he put them in a box and there was food outside the box and they had to push a switch to get the box to open. And he basically watched how, as they tried again and again, they would try like every possible thing to get out of the box until they happened by chance to press the switch. And then he tried it again and they learned and they did it quicker, faster, sorry. And they got better and better until they would know how to push the switch every time.
Mm-hmm.
So it would start with completely random responses of a cat trying to get out of something and ended up with one behavior being preserved
Well, that just
and being
proves
favored.
that just proves intelligence.
Wait, sorry, one second.
It proves.
1 second.
Okay, go
Wait.
ahead.
Okay. Now, that is how we later got everything we know about behaviorism. That was the beginning, the first idea of the operant conditioning, as opposed to Pavlov's work. And the whole idea of giving someone a bonus, a prize, positive reinforcement, it all starts with this cat in a box. Now, later Thorndike tried to do something else. He tried to get the cat to learn how to yawn to get out of the box. And that didn't work. What happened was cats, what he came up with was cats basically don't associate yawning in any way with getting out of something. So no matter how much you try to teach them how to yawn, even if they got out of the box, it wouldn't stick. They just couldn't do it. Whereas pressing a paddle wasn't a problem. So what he came up with was the idea that there is like a range of behaviors, and they are ordered according to what is preferable at any given moment, according to stimuli. And as things change, some of them become more preferable than others in reaction to the situation. And yet some of them will never, ever come up because they are that unlikely. They are that unrelated. So even though cats can yawn, they cannot learn to associate that with opening a box, not in any reliable way. And I thought of what you talk about, basic biochemistry, trying to create life and having selection, right? In those really, really basic. So basically what you're saying is that whatever might be close enough will happen first for some reason, like a probability or maybe there was more of one product than the other. Some 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 chemical response was preferable to another, right? That's do you understand where I'm coming from with this
Yeah, yeah,
line of thinking?
I can see what you mean.
So
Yeah. So from the perspective of typical psychology, what you just explained with the cats. I mean, for example, usually we used to think that the apes are more intelligent than the dogs, for example. Uh, that's what we used to think. But however, what it's appeared to be that some of the things that dogs can do, actually apes cannot guess it to do. It depends sometimes what is more suitable and what is more like embedded in the psychology of the dog.
exactly.
Uh,
So there's
for
always
example,
like there's like
things like a serving or helping, meaning giving something, like this stick game with the dogs. This
hmm
is quite very well understandable from the dog's perspective, because this is a herd type of animals. They are social animals.
hmm
Uh,
hmm
the apes also social, but in different way. So, and you can see it's very easy to learn to teach the dog to give you something from anywhere, to give it to you, than to teach ape to do that. So,
Wow. Yeah.
uh, but ape have different
So
ability. So it's kind of most interesting. It's a evolutionary psychology, I think, was called this type of.
exactly. And I'm trying to think of chemicals in kind of a similar way. Like this chemical has a certain pattern of behaviors. If it happens to meet up with that one, then it'll fit. And since there were more of those, then that was the more likely thing, right? So like in how behaviors are selected, I'm trying to model your selection of chemicals, see if I can understand it that way,
Mhm.
because I don't uh, like when you talk about how probable a reaction is, right? Because
Yeah,
some are more favorable,
yeah. Uh,
and yeah, sorry.
I mean, yeah, you can do an analogy with, for example, the neural system, how it's set up, and with chemical pathways, for example. But in
Mm-hmm.
the case of neural system, it's kind of it's sort of different and it's sort of the same. The difference is that if you take the brain as a whole, and if you look the activity of the brain, although I wanna first of all I wanna say I'm not psychologist. I'm not working in this area, but,
Mm-hmm.
but at least it's a basic principle. So the brain works in a fields. So you have neurofields in different areas of the brain, which is active
Mm-hmm.
or quiet, active or mocked. So, and
Mm-hmm.
those fields cannot be active at the same time. The same fields can, I mean, different fields cannot be active at the same time, or cannot be equally active at the same time. Let's put it this way.
Mm-hmm.
And that's how the brain works. It works in a fields, in an interaction. That's why. But what is more interesting, it's associated links between these different fields. So in the brain, and those links between the different areas of the brain is created by your experience, by what you
Yes.
set up to do. For
Yes.
example, let me give you some simple example. Sometimes you're going in the street and you smell something nice, right?
Mm-hmm.
You smell something beautiful, not beautiful, sorry, something pleasant. Uh, and that's remind you some moment from your childhood, for example. See,
Yes.
and this is how the links works between the different
So
areas,
what you're talking about is association,
association.
and we
Yeah.
have what we call Hebb's law. And Hebb was the neuroscientist who basically he looked at connections in the brain and said if a connection occurs again and again, it
Yes.
strengthens itself and it becomes more likely that it will reoccur. So,
And I assume the same is with the animals and any other brain
yeah,
containing
yeah,
creatures.
any synapse
So,
that yeah, like if it happened, it's more likely to happen again. It's the
exactly.
same association, and
So
that's all we are.
yeah. In a chemical world, when you have a simple chemistry or not so simple chemistry, the things are more like a straightforward. It's a preset. So it's like a Lego. You have a
Mm-hmm.
Lego blocks where you either build up something complicated or not. It depends on the environment. So
Mm-hmm.
that's how it is. It's based on
So
the law, it's based on the chemicals, attractions, and so on.
yeah, it's like if I'm building Legos, I also have to push it hard enough, right? Not just have the right two pieces, but also put in the energy to connect them.
Exactly.
So the chemicals also need the correct environment. And if we know the environment, we know which what is more likely to have happened, right? We can make better guesses.
It's not just guess. Sometimes the guess in the chemistry it's like 100% because you
Yeah,
know
I
the
know, I
law.
know, I know.
You know the
I,
law.
I'm using,
Yeah.
of course, it's chemistry. I know. What
So
I'm saying,
let me say, if you have acid plus base, if you combine acid plus base, what will
yeah, you
happen?
get salt and water.
You have salt and water. So
Yeah,
it's
yeah,
simple as that.
yeah. But what I'm saying is, we're trying to figure out what was the environment so we can guess at which chemicals might have been there until finally we will come up with the 100% answer, or not exactly 100% answer, but we will come up with the exact chemical response. Once we
Exactly.
have the right one, everything is a foregone conclusion.
And this is
That's
one
it,
of
it's
the
over.
yeah, this is one of the problems in the origin of life research because we don't know exactly what was the environment. We can guess,
Yeah.
we can hypothesize. It depends on the different environment. Sometimes we can say, okay, that was most likely the environment, but we cannot be absolutely sure.
Which is why I told Tangelo that the most honest answer is abiogenesis will probably come up with various suggestions, probably more than one, of how life might have evolved if conditions were so and so.
True.
And we will never know how life actually evolved on Earth.
Uh,
Because, as
we can,
as Ken Ham likes to say, we weren't there. I'm sorry, we might never know. We might never have the exact environment and always be stuck with two options. It doesn't matter. We're trying
uh,
to ask how it might happen, right?
yeah, so what we can do as a scientist is just to uh, recreate the life in the modern conditions. However, science always surprised the people, I mean scientists. Sometimes we might find something is a very clear evidence how something happened.
Yes, definitely.
Not to forget, I mean, it's coming a huge research after all, but also we are looking the neighbor planets at the moment. So we might find evidence in another planet that the conditions of the Earth was like that, and that.
Yeah. If it's similar enough and younger than us.
Take a piece from Mars, for example, and put it in a specific conditions in Earth, and we might observe some very interesting interactions happen there.
Yeah.
So,
and that's the thing. It's trying to solve the riddle and not necessarily getting the 100% answer of this is exactly what happened.
And unfortunately, no, there is no scientist at the moment that can say that. I mean, nobody can say that. Even if we have a very good hypothesis, the hypothesis serve. What is the idea for hypothesis of origin of life? The idea is to have something like a preliminary to know what we are looking for, because you cannot just go and dump a bunch of chemicals in something and waiting for rabbits to come out.
Yeah.
So that's not the way how the science work and the origin of life science work. So we have to approach somewhere and somehow, and you have to create a hypothesis which is, although you know it's not exactly that happened, you have some approximations what you're looking for when you're trying to go and try to do experiments. Because experiments, it's coming, almighty experiments. This is the king, almighty experiment.
So I'll let you in on a secret. I have a friend. He is he's working on his doctorate in agriculture, and he's a very, very intelligent person, and he studies this like alien fungi. He's a very interesting guy. And he told me that after working with actual living systems, as in trying to figure out how the fungi actually works, and he said, I don't believe a word of any kind of science that does not happen
in front of me.
What do you call it? In vivo or in vitro? I keep forgetting.
Yeah.
Like he says, if you study a protein outside of the organism, I don't know, because you might get anything.
Well, we study all the time in vitro, in vivo things. So
yeah, so I'm just saying about abiogenesis. I'm trying to say that we're trying to figure out how the principle, right? First of all, of how it might happen, right? That's the first step.
Yes.
And okay, so you thought of a principle, and then how do you move from that?
So you have to figure it out what actually you're looking for, because
which chemicals?
Yeah, so not just chemical. What kind of interactions you're looking for? Because you can see it and might not recognize it. For example, I can give you an example for that. Back in the 90s, one of the pioneers of early research of origin of life, Orgel, he and his colleagues, of course, they provided some experiments which are based on RNA world experiments. So back in their heads, they have RNA world hypothesis. And based on this hypothesis, they set up experiment where they try to figure it out, is it possible RNA to replicate without enzymes, without any needs of extra activities other than nucleotides and the actual RNA. And based on this experiment, non-enzymatic replication, they actually figured it out that in fact the replication occurred, but it occurred only on a G and C nucleotides. So, but once you add adenine and uracil nucleotides, type of nucleotides, the reaction stops. See, at that moment, they consider that as a failure of the experiment. They consider it as a failure because back in their background was RNA world hypothesis. And based on the RNA world hypothesis, you're supposed to have high fidelity, all 4 nucleotides, non-enzymatic replication.
So basically what they found, if I want to stop you and understand, because again, I'm very lay, like a lay person in these fields. You're saying that they had an idea of RNA would self-replicate, and they found out it couldn't, and so abiogenesis supposedly crashes. Because the RNA world is not possible. So supposedly, allegedly.
So it's interesting because you're approaching your experiment. You're doing your experiment based on the idea that you must have, according to your hypothesis, you must have a relatively high fidelity and all 4 nucleotides, non-enzymatic replication. So there's no proteins, nothing else, just reaction. And they found that it's not possible to have high fidelity and all 4, but it's possible to have mainly GC incorporation on the template of the RNA. So replication occurred. It's possible to occur,
but not high fidelity.
Not high fidelity. It's not with
lost some of your chemicals.
Yeah, just with G and C. And guess what?
The replica was simply missing certain compounds.
Yeah. So my point is here like that. So during that time, they considered that as a failure of the experiment. Because they thinking that you have to have different types of they think during that time that you should have all 4 nucleotides high fidelity, but didn't have. And now, just now, it's happened to be based on a modern hypothesis that actually this experiment is absolutely valid experiment, and also clearly shows that it's not a failure, but contrary is actually what might be indeed happened. According to the new hypothesis, new scientists' hypothesis, like RNA peptide world, not RNA world, this is good enough. This is good enough to kick on and to continue origin of life.
So let me stop you for a second.
Yeah.
If I'm understanding correctly, our new hypothesis, which talks about a simpler, shorter version of RNA, says, wait a minute, 2 are enough.
Yes.
We don't need the high fidelity of 4. We just need high fidelity of 2. So an experiment that got us those 2 is a knockout.
It's a good experiment. And in fact, it's a very nice experiment. So, but during that time, they didn't recognize that it's a good experiment. They thought that it's a failure because it was coming from
it didn't match their expectations.
Yeah. So my point was that's why we are building up different hypotheses, because we have to figure it out what it's more logical, what it makes sense to looking for during the origin of life. Not just going and try to play and waving and put chemicals and boil it and try to figure it out, basically nothing. So this is the good way of science, and it's not just origin of life. Every science, good science, start with observation, build up a hypothesis, you have some prediction for that, and do experiment. So that's how it works.
Yeah, and if only people would understand. There was something I wanted to ask you. Otangelo keeps bringing up his really weird ideas, and a question he's asked you a million times, and I want to have another go at this, is like...
I just showed one.
Sorry, what?
I just put one of his weird ideas on the screen.
Okay. So he like says that DNA is like you have a dictionary. You have an assignment of meaning. And we keep trying to tell him that there is no dictionary. That the dictionary is an intermediary step for intelligence like us. In chemistry, that's not there. There's just the actual reaction. No one knows what it's doing. It just is. Am I making sense here?
So I'm not reading Utengelo's, but whatever you said, let's try to understand first, to be clear, what he means by DNA is a dictionary. I mean,
what he means is, okay, you have the simple idea that there is something outside and there is a symbol to signify that something. And the dictionary is a table or a book or a Rosetta Stone containing the assignments, telling you how to connect symbols with actual things, or how symbols are used.
This is okay. It is correct and it's not correct.
Okay,
what I mean. Yes, indeed, the information is written down. Let's say this way, written down on the DNA. So the DNA is recorded. Everything is recorded there. However, if you think about what translates this information into function, there is two things that translate into function. One thing is the genetic code, which translate, and the other is Watson-Crick base pairs. Therefore, if you don't have, let me say, you build up different genetic code and put it with the same DNA, with the same sequence of the DNA. Immediately, if you have different genetic code, this sequence of the DNA will not be able to be translated at all. So in that case, you must have a rule for reading. Without the rule for reading, everything that is written on the DNA, it's not valid. It will become gibberish.
Okay.
So, and because there is a rule of reading, the DNA makes sense. The actual what is written on the DNA, it makes sense. So to say that it's a dictionary I wouldn't say exactly dictionary, because dictionary translate from one language to another. Here you have two languages. One language is the base pairs language, and the other language is the protein sequence.
Yes.
So the translation between RNA or the DNA sequence and the protein sequence, it's happened in the process of translation, which
Yes.
is the process of protein synthesis. And that's entirely
Okay.
based on a genetic code. And
Now I'll
that's
stop you. I'll stop you for a second. And here's where Ontangelo comes in and says, aha, you said translation. Translation requires someone who knows how to translate.
yes,
Someone has to know the languages.
yes.
And Otangelo wants to know who that is.
I can tell you.
Who
So
again, sorry?
So who knows how to translate? There is only one type of enzymes that actually knows how to translate. And this enzyme, it's called amino acid synthetase. You say, who knows how to translate? The answer is the amino acid synthetases are the actual proteins that knows how to translate.
Okay, what does it mean that they know? Who taught them the languages? Did they go to school? Do they have books?
Yes, I will explain. It's embedded in the structure of the peptide. So you have a specific 3D structure of the protein, which is capable to recognize two things. Mainly two things is recognizing. One is the amino acid, and the second is the anticodon of the tRNA.
And recognize just means that they react with each other, right?
Yes. So
it's no more profound than that, the word recognize.
Recognize in a chemical way mean it interacts.
And if something else comes along, they just don't interact. Right? Nothing happens.
Nothing happens. If you don't interact, nothing will happen.
Right? So all kinds of chemicals move along, not interacting, meaning not being recognized, until the right one comes along and is recognized, meaning there's a chemical reaction.
So, simple answer to the question, who knows the genetic code? The answer is amino acid synthetases. So, why is that? So, to be able to recognize the right amino acid and the right anticodon at the same time, and to put them together in the tRNA, this is what is the genetic code knowledge is. So, meaning to link the right amino acid with the right anticodon. This is the genetic code.
So it's simply another chemical that reacts correctly with what we need.
Exactly.
And there's no more to it than that.
No, that's it.
They're not talking. He's not saying hello. No, the one chemical has not been ordered to arrive.
No.
Right?
Even there was a recent, I remember in probably 5-6 years ago, there was an attempt to put a new codon and new amino acid in the codon just by redesigning the amino acid synthetase. So if you redesign the amino acid synthetase, you will be able to make a new amino acid and new codon to put inside. So it's simple as that. So
yeah, you just change the chemical. You put in a different chemical.
Yeah, I mean,
or maybe just fold it differently. Just
exactly.
Change its shape.
And of course, there are lots of details how it recognizes, what exactly it's recognized, but always it's based on a structure of the
Yeah,
not
but
structure and the charge of the actual amino acid synthetase.
what is important for me to get across is that when we use the word recognize, it's easy, simple language.
Recognize
Right?
it's a fit with the structure with structure. So
exactly. And if you were teaching it in class, you would just write an equation. You wouldn't even have to say recognize. You would just show them the reaction and what product we get. That's called recognize.
Yeah.
Right?
Yes.
And
yeah,
I think use that.
I think, Meny, you have made very good questions in this entire sequence, and you actually are understanding what the genetic code does, which is something which Leo Filius questioned and claimed that it was um, metaphorically a code, but not in a literal sense. And what you described is precisely what happens. There is a translation process where the codons are assigned to amino acids. And when a specific codon goes into the messenger RNA, the codon sequence goes inside of the ribosome, then the tRNA, which has the anticodon, which matches to it, is selected, and then the amino acid tRNA synthetase picks up and charges the correct amino acids and attaches it to the tRNA, which then elongates the polypeptide chain. That is the process.
Otangelo?
Yes.
Can I ask you, please, walk with me here, okay? If you have a square-shaped hole, okay? And you have all kinds of pegs. Some are triangular, some are cylindrical, some are hexagonal, and one of them is square. And you try to push them one after the other into the hole. How amazing is it that the square goes in and the others don't? How incredibly amazing is that?
Well, your analogy fails on several accounts.
No. Answer my question. Don't tell me how it fails. I'm asking a simple question.
That is
There's
not
nothing amazing about it.
That is not a good question, but okay.
It is a wonderful question.
Okay.
It is a wonderful question.
It fits.
Because
the one which has the correct
if you don't want to answer,
okay, so
Otangelo, you can say I don't want to answer your question.
No, I just answered
but
it.
Please don't change it.
Okay, so I just answered. Of course, if you have the matching form, then it will fit and it can go through.
Great. Is that amazing? Is that spectacular that a square peg fits in a square hole?
Why should it? There is nothing special about that.
It's not spectacular in any way.
So
why is it spectacular that one chemical fits with another chemical?
Because it should.
Why is
there
that
is no
so
reason.
Spectacular?
Well, because there is no reason that it should be so.
Yes, there is. One is a square hole and the other is a square peg. That is the reason. There is a reason.
Okay. Just
That's
one second. Can I explain something? First of all, don't talk over each other. So be patient. The second, Otangelo, as far as I understand, you agree completely that if you have structure A and structure B, they might fit each other, right?
Yes.
So you agree with that?
Of course.
What you don't agree that there is no reason those two to stay together, or what exactly it's you don't agree after that moment?
Well, I mean, I will try to exemplify and to make an analogy in order for you guys to understand. I am a machine designer. When I have to make a machine, where the individual parts require to interact together and to fit together in order to have a specific function, then I need to use my intelligence with a specific goal to make these parts in order for them to fit together and to exercise a specific function.
Okay.
And I do not know of any random unguided mechanism which has this
Tangelo.
capacity to.
Just one second.
Tangelo,
I would like to finish my point.
Let's
sorry.
Let's finish.
Sorry, go ahead. Sorry, that's me. Sorry.
So I try to simplify as much as possible to attempt to make my point clear. There is no natural, non-intelligent urge or goal to make specific things far ahead and then to make machines which accomplish that goal further ahead. And in order to have a specific outcome further ahead, made by a machine, you need intelligence, and it's special when that machine which performs that action in order to have a specific outcome is dependent on several interacting matching parts. And that is exactly what we see in the ribosome, where you have a variety of different interacting parts, and you have the genetic code, which is literally an assignment of meaning of the codons to the amino acids. And that is a problem which I think is a conceptual problem, because we do not know any example of unguided natural mechanisms being able to assign meaning.
Okay, so Otangelo, listen. The answer, you keep saying it wrong. Nature is nothing like a designed machine. It's nowhere near that. Nature is like the Big Bang Theory. Nature is like how stars are born from dust falling into each other. Nature does not look like a designer sitting with a blueprint. No one intended it. What happens is, for some reason, something begins, something is added onto that, and then something is added onto that. And you want to look at something way, way down the line, and you say, oh my God, whoever started this process must have known how it would end. Otherwise, how could he have started it? That's basically what you're asking, right?
Well, first of all, we are not talking about stars and the universe. We are talking about molecular machines, which are machines.
We are talking about nature, how nature behaves. Nature does not behave like blueprints and design towards goals. It just doesn't, ever, anywhere.
Okay, Otangelo.
High speed.
Have you ever seen a machine that has two gears and it works by the gears flying around at 50,000 miles an hour, randomly slamming into each other? Have you ever seen a watch work that way? Is that a good description of a machine?
Of course not.
Okay, then the cell is not a machine.
Well, I'm not claiming that the cell as an entirety is a machine. What I am claiming is that the cell hosts many factories and molecular machines.
Many.
I am talking and you are over talking again, and I would like.
But you're repeating yourself. You're just repeating what you said before.
Bye. I mean, Meny, when you can have a dialogue without interrupting, then I would be more than happy to come back and having
I'm a very limited human
conversation.
being. Have a nice day. Bye. I'm sorry. It's who I am. It has nothing to do with him.
Yeah, it is difficult to listen to the same argument over and over again, or the same.
He was just starting to repeat himself. There's no factory.
I know.
It is not. It's like if I dump poison. It's you know what what he's asking about how could it have intended if it's nature? Of course, nature does not intend anything. He's trying to say that I could not be here unless my great, 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 great grandparents could see the future.
Yeah. There's
how could they have planned me?
There's
right?
There's this confusion of causality. There's an example in this biosemiotics book. Like if somebody comes up and rings the doorbell, and you hear the doorbell, then that person has sent you a message that they want to talk to you. They want to give you something. They're at your door.
Yes.
And so we look at the person ringing the doorbell as the cause of the message and you as the receiver of the message. But suppose you have your headphones on and you're listening to Pink Floyd at high volume.
Then there's noise.
You don't hear the doorbell. But consider that that is not the only cause of the doorbell. There has to be electricity. There has to be a doorbell. There has to be, all these different parts put together.
Air.
Somebody put them together. So like you can try. So what it is when you look at the system as a doorbell, you are coming up with a prescriptive description. So you're describing a prescriptive process. And you make the mistake of thinking that is the final cause. And there is no such thing as a final cause. Mm-hmm. Everything works in this massive network of causation. And you cannot find, And so we reduce our biochemical pathways to things that look like Boolean logic and they look like machines. But this is our reductive mind trying to understand. And that is the problem. So we basically let our reduction get in the way of our intelligence. And that is what D'Angelo is doing. So
he thinks that the reason
exactly
a certain protein and a certain whatever happen to fit together, that means someone built them that way, because otherwise nothing would work.
Yeah.
And it's the other way around. That worked because that's chemistry. And then whatever happened next is life. Life grew around what worked. Like what worked chemically gave rise to what works biologically.
There we go. Let me see if I can make it bigger. Is there a plus?
Okay, guys, I'm sorry. I'm going to have to go now.
Where are you going to go?
I have a life.
You have a what?
Basically enough. A life, yes.
I have a few hours left. I'm hoping that before I go, I can get to 200 subs. But it's looking bad. I need 7 more subs, and the old ticker is starting to palpitate. Anyway,
guys,
okay, Meny, thanks for
stopping. Give a like
stopping.
and subscribe, because speed might not live.
Yeah, might not make it to the end of
pity.
I might break my hip on the way upstairs. So
God.
Okay, thanks for stopping in, Meny.
I'm gonna cry now. Bye, everyone.
Goodbye. Hi, Nestle. So I got your paper up here. Is the cell really a machine?
Oh yeah, I cannot also stay here for long because I need to study more for my exam. So.
Oh, what do you study? What is your exam on?
Plant biology.
Ah, so is that your field? Plant biology?
Yeah, yes.
Plants. You see, we don't pay enough attention to plants in these damn evolution discussions. So we need more plant biologists, because I think plants are really, really easy ways to see evolution happening.
Mm-hmm.
I don't know. And they are just absolutely amazing. And they're such different critters in some ways than animals, plants. I like the fact that the reason I like to study plants is because they sit still. Fucking bugs don't do that. They fly all over the place. Anyway, we have a paper to look at.
Yeah.
That's like, you got any questions for Demeter or?
So I just wanna actually once and again, to answer to Otangelo, because this is his very frequent argument, and he always wanna say that the molecular interactions or the chemistry does not have urgent to make life. Or I will say no, it doesn't. He don't see how, if something is a sequence, if there is information, he don't see how the natural urgence for that information can occur. That's why he used to say, Otangelo, he used to say that when you have set up something like a cell or cellular interactions, he don't see the needs for this. And this is his argument all over and constantly, again and again. And unfortunately, he quit the live chat here. But if he come back, I can explain to him anyway what I'm talking about. So, Otangelo, you can come back. I can explain.
Yeah, where did Odie go?
Many?
No, Odie.
Oh, Odie.
Instead of calling him Otangelo, I'm shortening it. Now I'm just going to start calling him Odie, O-T-I-E. I had a question for him, and that question is, have we ever observed an intelligence design life?
Yeah, I know. That's what I asked him last night over and over. And there was...
So let's explain Otangelo's question. Can I put a paper here? So, Mike?
Yep.
Let me share. So let's see how
I was going to look at this paper a little bit.
Okay, I'll try to share my screen.
There, you're on camera. Now you might hit 200 subs.
Not the way I look. Not with those freckles on top of my head. I gotta get rid of those freckles.
Okay, can you see my share?
Yeah, there it is. Okay, we got it.
So
can you link that for us too, so that we can give people a link?
Oh,
link.
I have to go to NCBI.
Oh no, okay, I'll do it. You go ahead and talk. I'll do it. I'll find it.
You can find easily in NCBI.
Yep.
Just go and see. So this is one of the recent papers from a team from Loren Williams, and as you see, there are lots of people here. The actual paper. First of all, I just want to say that the Loren Williams is one of the first guys that presented RNA peptide world as a more feasible origin of life hypothesis than just RNA world. And based on this hypothesis, everything started with the interaction between the RNA and peptides. So here, particularly in this question, as experiments, we're talking here for the real experiment, we see how if you have interactions between short peptides and RNA, the stabilization of the entire complex became more stable. It is more robust and it's more difficult to destroy, let's say this simple way. And all of the characteristics of this peptide RNA interactions shows clearly approximately several times more stabilized complex. And now I can say it this way. If you have a peptide RNA interaction, which it shows experimentally to be more stable, and if you have environment which have harsh conditions, heat, acidity, pH, all kinds of chemicals, and so on, based on this experiment, you will see that if you have interaction with RNA and peptide, you will have more stable form. What does it mean, stable forms? It means that will stay longer in environment. Is that correct? Am I crazy or not? So it makes perfect sense. If you have something more stable, will stay longer. In the,
Yeah, and that is a selection function.
so this is the selection. However, also you have to pay attention that not every amino acid is responsible for peptide RNA interaction. So as a part of the peptide, if you have some positively charged amino acids like arginine, lysine, histidine, or many others, several others, which are not canonical, some of them are not canonical, you will see that these interactions will happen because of the specific amino acids as a part of these protopeptides, of these peptides. And particularly in that case, they clearly investigated these features. So, in order to say that there is no,
I have a really important question.
there is no anything that force nature to produce life or to produce information. Actually, this is the force. This is the urgent. It's simply if you have a specific interaction which is more stable, absolutely for sure you will be there. Therefore, you have selection of those particular sequences which are responsible for these particular interactions. And this is what this is what is the necessity that Otangelo is trying all the time to say that there is no necessity. Yes, the necessity is simply the stability of the process. There is nothing else. So, simple question, simple answer. Yes, there is a necessity. Usually, Otangelo used to say, if you see the sequence, I don't see what is the necessity of this sequence to exist. I mean, the necessity is the survival. The necessity is the actual stability of the complex. That's what it is all about.
Can I ask a quick question?
Yeah.
How many people work in a cell?
People.
I only ask that because factories, by definition, have workers. So how many people work in a cell? What time do they clock in? What time do they clock out? How is the hierarchy structured? Like, do they have bosses? Yeah, how much pollution does the average cell give off? That might be what's causing climate change, because there's so many goddamn cells on the planet. And I mean, if they're factories, they've got waste.
Well,
so.
I mean, the oxygen is pollution, actually, from the perspective of green algae. So, green algae polluted with oxygen our planet. So, but that's not the point. I mean, I can see your sarcasm a little bit.
Yeah.
But, so, I mean, this is all about stability.
Icarus asks, do cells have unions? Yeah. The workers are they unionized? I'm sorry, I had to.
Angelo put up a book called The Molecular Fabric of the Cells, but then he said there were two texts, and I can't find the other one. So, but I don't know about this book. Have you looked at this book? Molecular Fabric of Cells by who wrote it? Let me see. Carell?
Steve Carell?
No, not Steve. B.C. Carell.
Oh, Curl. That's probably Curl. I would have.
Corral.
Maybe it is Corral.
Corral.
I don't know.
Corral.
I'll be back.
Loki, I'm having a problem, Loki. I can't move my mouse with you sitting there. You have to get off my mouse.
Can he?
Loki,
Can he hear?
Yeah, he can. He can hear. Meow at him.
Meow.
He's
Meow.
looking.
Kitty, 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 kitty.
Loki, Loki. Say Loki.
Loki. Loki. Male or female? Male. I'm assuming.
Male. He just moves his head a little bit.
Kitty, kitty boy. He's probably like, what is that? Where's that coming from?
Yeah, they don't. They learn to ignore most of what I listen to, and but it's amazing if I have animal shows on, he gets all excited. Like I had this one movie about a fox. The whole movie was about a fox, and he was just all excited. He was following it. But then if he sees horses on the screen, he goes nuts. He thinks horses are the coolest looking mice. So he'll sit and watch an entire documentary if there are horses in it or a movie. I don't know why he loves them so much. I don't know. Do horses look like mice? I don't know. Anyway. Okay.
Just a second here.
So, let's see what else.
Jet.
So Tangelo's going on about, okay, you guys keep making the same basic questions when I answer incorrect. Oh, he's correcting us. Let's see. And when I answer incorrect, you guys accuse me of being repetitive. So we're making the same. Yeah, we're telling you that the cell is not like a factory. The cell.
Well, I would say it's, analogically, yeah, it's kind of like a factory, but obviously not in the literal sense. The same way that
some cells,
you can say that a star is like a factory, but a star is not literally a factory.
Right,
there aren't little elves in there forging elements. Hi ho, we're changing this to helium from hydrogen. No, that's not going on. It's not the same thing. It's not happening in the cell. It's literally just a bunch of advanced chemical reactions taking place in a specified manner, producing particular outcomes. That's it.
Well, yeah.
So very frequently about the origin of information. So let's discuss a little bit about origin of information. Biological
information in general?
No,
biological
information. In general, I wouldn't say, but biological information it's a result of selection. Again, with the same principle that I just mentioned. If you have peptide RNA interaction and this survives better, and if this interactions depends on a specific sequence, of course that sequence will stay. It makes perfect sense. Once you stay this sequence, this is the information. So what is information in biological meaning? Information is a specific sequence of something which gives a function. Simple, right?
Yeah.
If you have function, you have information. If you don't have function, you don't have information. So, based on this, information always gives a function, and the function during all the times is some sort of survival. So, in the very beginning, life complexes are more stable, based on some specific sequence. So once you have a specific sequence, you have stabilization of this sequence, leading to survival, which is the function. Here we go. We have, in that case, origin of life, origin of information, and origin of structure, origin of function. I don't understand what is not understandable here. Why Otangelo keeps saying that there is no possibility to start information. This is the information. Stabilization. Some sequence simply leads to more stable complexes. That's it.
Yeah.
I mentioned earlier, for example, the experiments of Orgel in the 90s, which he showed that replication is possible, meaning non-enzymatic replication. There is no enzymes. It's just like a chemical rearrangement of the nucleotides over a template. So you have a template RNA, single-strand RNA, and you arranged the nucleotides on the top. This is happening naturally, chemically, but during these very early experiments, that's happened mostly on G and C nucleotides, but not on A and U. Here is the selection again. You select versus G and C in a completely natural, uh, process. So that's one thing. Also, what else? In the origin of life, right now, we are in the kind of era, scientific moment, scientific situation where everybody is talking and nobody is listening. So how it's called that? So there is a
Sorry.
yep. You
Spooky sounds.
yeah. So Leo,
Yes.
about the so-called self-replication. It's a bug. This is a modern bug in the science field of the origin of replication. So first of all, let's specify what is self-replication. There is two aspects of self-replication. If you're talking about cell, like it is like a cell, it is self-replicate. So you can completely legitimate, you can say, yes, this is a self-replication of cell. Yes, that exists. Second, you can say, well, if you have some sort of system between RNA, DNA, RNAs replicate by proteins, the proteins are synthesized by RNA, there is a system, and those systems can self-replicate as well. So you can say, yes, there is such a process of self-replication of the system. However, when you come to the origin of life based on the RNA world hypothesis, the idea for self-replication is about replication of RNA by RNA. So you have a ribozyme, which is completely by RNA, and replicate itself and makes another ribozyme, which is replicase or polymerase, but it's entirely built by RNA. So this is a self-replication on an RNA level only. And here is the moment. The problem with this hypothesis for this self-replication on RNA level is because this ribozyme entirely built up from RNA is too big, too long. And if you chop it a little bit and make it smaller, it will not function. This is sort of creationist argument for irreducible complexity, sort of. Meaning you don't have a smaller ribozyme that can perform polymerization process. You have to design it intelligently and to build up this ribozyme. And actually that's what they did in the 90s. So they created such a ribozyme, which is self-replicating ribozyme. However, during the origin of life, those processes are unlikely to occur. First of all, by RNA peptide world hypothesis, we don't need this process at all. Self-replication ribozyme, it's not necessary. To me, this is a dead end of the hypothesis. Despite the fact that yes, indeed, has been created in a laboratory condition. So from the perspective of ribozymes, you can make pretty much everything that you want as a function from ribozymes in laboratory by process of selection, by process of that. However, it's very unlikely that to happen naturally. So, ribozymes, natural ribozymes, which actually do exist, ribozymes, and they're responsible for several very important functions in the cell, like processing of tRNA, like, for example, self self-splicing ribozymes, also a so a specific cut of the RNA, so split the RNA in a specific place. Those kinds of ribozymes actually do exist, and this is a natural ribozymes. But all of these ribozymes usually works with the ligation steps, so interaction with the RNA and making a specific structure to make either a recombination process or just a cut. However, in the nature, naturally, there has not been observed at all natural ribozyme which is capable to polymerize RNA. So usually when I was with Ron Ra in just discussions with Ron Ra, he mentioned, yeah, there is such a paper. Well, there is not such a paper. Whatever has been given to me actually shows that, for example, that was with hepatitis virus. So the hepatitis virus it's an RNA virus, but does not need RNA polymerase, meaning does not need RNA dependent RNA replicase, which is available. Those kinds of polymerases exist. This is a protein polymerases. For example, COVID-19 have this type of polymerase to amplify its genome. But those RNA viruses actually do a replication using a cellular replicases, like a polymerase 1, polymerase 2, which is responsible for transcription, for transcription. So it's not the same. It's not like a lacking of any protein polymerase and self-replicate. That's never happened. And it's not observed at all in the nature. So based on RNA world hypothesis, I think this is a dead end from RNA world hypothesis. I don't think ever happened self-replication on the level of RNA. It's ever happened at the beginning of life or ever happened after that. So, but if you look about the replication, because this is a major problem in the origin of life. It's like a stone, which you need to explain how the replication occurred. You must have some explanation how replication occurred. In fact, it's very easy to explain from the perspective of RNA peptide world, because all polymerases that polymerase RNA or DNA, they possess 2 or 3 magnesium atoms, ions actually, not atoms, ions, which stay in a specific position. And this is the catalytic center. But those kinds of things could be done from polypeptide. So simple polypeptide could facilitate those processes very easily. I mean, not easily by meaning easily, but explanatory, explanation type of easily. So this is the one of the, everybody when you're talking about origin of life should understand this, this moment. You can explain replication very well by RNA peptide world, but it's very difficult to explain some sort of large self-replicating ribozyme. The problem here, the problem is that it's still RNA world, those type of hypotheses, it's still very it still stay in the minds of lots of scientists. It's still circulating the science. It's still, you can see lots of papers that continually repeat self-replication, and I think it's a wrong. So that's one very important moment. What else?
I don't know. Where'd Speed go? He's usually the one that's asking the questions. I have no idea where he's at.
I'm kind of here.
So from the perspective of probability of origin of life, it's very frequently, especially creationists, they used to say that it's highly improbable to have such a big protein appeared because of blah blah statistic and so on. And we have to trust the statistics, right, Leo? So the statistic is a powerful method. There is only one place I never see in statistic to work well. Early in the morning when I'm looking for my socks, urgently going into work, and I'm trying a huge pile of dresses and so on. Early in the morning, I'm trying to pick up my socks. Statistically speaking, you always have to have high probability to find the same socks in several picks up. Unfortunately, that's never happened. Every time when I'm picking my socks, a statistic breaks completely. So you cannot find the right socks all the time.
Well, socks notoriously break the rules of
exactly.
physics.
So
like when they disappear in the dryer,
they always disappear. And very frequently, I'm going to work with two different socks because there is no way to find the same one, the same type. So, and this is the moment where the statistic completely breaks. So, but other than this particular moment, always works well, especially in the number of amino acids that you have to order in order to produce protein or something like that. So let's assume that somehow by low, super low probability emerged very well developed protein. And there's nothing else around. So what will happen with this protein? Nothing. This protein will emerge and will completely destroy by itself, by the forces of nature will destroy it, and nothing will happen because this particular protein, it doesn't matter how big it is, there's no function for it. There's no surrounding for this particular protein. So and that's usually how wrong is the thinking when you try to figure it out, the origin of life by statistics. First, I've never seen any scientist claiming that you have to develop completely well-developed proteins. That's not possible. You always start with a short peptide, which evolved in a longer peptide, and the longer peptide evolved in a longer peptide. Finally, you have protein. So here I'm repeating again the same thing which everybody knows by now. The difference between peptide and protein. The peptide is short. It interacts with whatever amino acid it contains. So if it contains positively, will interact with negative. If it's hydrophobic, will interact with hydrophobic. If it's blah, blah, and those kinds of things. However, once it became longer, it starts to fold. You have a folding process. And once you have the folding process, you create the catalytic centers. You have motifs, you have domains, you have catalytic centers. Therefore, you can bring different functions now. The peptide by itself have a limited number of functions, limited possible functions with the composition that they have. But the proteins, which are folded, they have unlimited functions. They can be everything that you can you can imagine. It depends on the selection. That's why the evolution goes from peptide, motifs, domains, full protein. And it is simple as that. And that goes in a co-evolution of everything. It's co-evolve. So this is the answer for most of those kinds of questions that usually the people. And I thought Mike, Andrei Lupus is the guy for those kinds of representations. They have a good papers showing a specific small domains or motifs and so on. Mike, if someone wanna come and ask questions.
Yeah, we only got let's see, we only got 4 people watching.
Okay.
We're up to about 2 hours.
Yeah, good enough.
Yeah,
So-
it's a little after 12:30. I got to take off because I got some
Yeah.
stuff that I got to get done
Okay,
today.
so let's stop for now.
I like keeping my streams less than right around 2 hours because YouTube won't give you transcripts.
Because you're a pussy.
Yeah.
Yeah, that too.
You're a little pussy baby streamer.
I'm a little baby streamer. Yeah. Boomer streamer.
So this after probably around 3-4 o'clock, if you're streaming again.
Yeah, we might do it again.
Around.
Are we doing Snatta tonight?
I'm hoping tonight or tomorrow night.
I'm hoping tonight. You know Snake was right, Speed?
Yep, he just subbed to me, by the way.
Okay, because I told him that if he wants to join, he can. So he said that he's had some email exchanges with you before. Do you have his email?
Yeah, I'm hoping I still have it if it comes.
It's literally just. Well, I'll put it in the side chat that way everybody listening can't see it.
Snake. Let me see. No, it doesn't come up as Snake. So
that's because I put it in this. Once it loads and sends the fucking. There it is.
Okay. Yeah. Okay. Why the hell didn't that come up? That should come up.
I don't know.
Okay.
Oh, Mike,
anyways, I'm out
Mike,
here, guys.
I want to talk something
see you later.
with you, but after the chat, so you can close the chat and
Okay.
I want to talk with you.
Okay, I'm going to end the broadcast, everyone. So thank you all for showing up. Ending now.